Okay, hi, this is video C of Vestibular Function 1. And in this video, I'm going to be talking about linear acceleration and how our vestibular system picks up linear acceleration. So there's two main types of linear acceleration that we can talk about, and then there's parts in between. But we can talk about horizontal linear acceleration. So like my Superman here, I had to look a long time to find a Superman with his head like this, but his head is vertical to the horizontal plane, and so he is moving and accelerating horizontally. And that's going to be activating one of the otolith organs, which is called the utricle. So the utricle is responsible for picking up horizontal acceleration, linear acceleration. And then we have another otolith organ, which is called the saccule. And the saccule is responsible for picking up vertical acceleration, so going up and down. So this guy here rented this thing to go propose to his partner. So as he was zipping up, I just noticed he has bare feet. I had noticed that before. As he goes up, he's getting activation of those saccule hair cells, and as he's going down, he will as well. So how do these otolith organs detect this type of stimulation? So it turns out that otolith organs are very similar to our semicircular canal organs in that we have hair cells, and we have hair cells that are connected by tight junctions. Our hair cells have a hair bundle with this typical staircase-like arrangement of these stereocilia, so taller ones down to shorter ones. Just like in our semicircular canals, we have the apical parts being bathed by endolymph, and we have the basolateral parts being bathed by perilymph. Similar to what we've already talked about, if we move the hair bundle towards the tallest stereocilium, we're going to get depolarization. If we move it away from the tallest stereocilium, we're going to get hyperpolarization. And these hair cells in the otolith organs are still going to be releasing glutamate. They still have baseline activity. So when the hair bundle is not being moved back and forth, there's a little bit of glutamate coming out. Depolarization is going to increase that glutamate. Hyperpolarization is going to decrease that glutamate. And they are going to talk to specific fibers that are in cranial nerve 8, which is the vestibular cochlear nerve. So a lot of similarities there. So one of the differences is that these hair bundles are in a gelatinous mass that has these little calcium carbonate crystals in it. So these are calcium carbonate crystals. And these calcium carbonate crystals, together with this gelatinous mass, are called an otolith membrane. And the otolith organs are named for this membrane, and they are named this because oto means ear and lith means stone. So these little calcium carbonate crystals are referred to as stones. And this otolith membrane, these little crystals make it more dense. So this is going to be increased density relative to that surrounding endolymph that's surrounding it. So what happens is if we get movement, and what I've drawn here would be actually the hair cells in the utricle, because if we get horizontal acceleration, either this way or this way, that is going to end up affecting this otolith membrane such that it's going to change how much this hair bundle is bent. So if it's bent towards the tallest stereocilium, it's going to depolarize. Bent away, it's going to hyperpolarize. And so that's our utricle, which detects horizontal acceleration. It turns out that our saccule those hair cells are oriented a little bit differently. So this would be our saccule, which is going to detect vertical acceleration. And those hair cells are going to be oriented more like this. Same characteristics, though. They're still going to have tight junctions between them. They're still going to have endolymph bathing the apical part and perilymph bathing the basolateral part. They're still going to have an otolith membrane that's going to have those calcium carbonate crystals in it. But now, if we have vertical acceleration or deceleration, that's what's going to activate these saccule hair cells. So vertical acceleration. So this otolith membrane, if we look at it with scanning electron microscopy, this is what it looks like. So this tissue has been fixed, so there's holes in here that wouldn't normally be here. But what you can see is these calcium carbonate crystals, so they're quite prevalent. They're embedded all over this membrane. And then you can also see these little hair bundles poking through here. So you can even see the size of the crystals relative to those hair bundles. So even though they're really very small, relative to the hair bundles, they're quite large. They're going to, again, increase the density of this membrane, which is going to affect the hair cells when the head is accelerating, and also when the head tilts, because they're going to be more dense than the surrounding endolymph. So it turns out that these hair bundles, the orientation, so which way that talus stereocilium is oriented, changes throughout the organ. So here we have the macula. That just means the sensory epithelium of the utricle. So that's the one that's going to detect horizontal acceleration. These arrows here, the tip of the arrowhead indicates where the tallest stereocilium would be, and the base of the arrowhead is going to indicate where the shortest stereocilium would be. And you don't need to memorize this, but what you do need to notice is that no matter what direction in a horizontal plane you're moving, some of these hair cells are going to be depolarized, some are going to be hyperpolarized, and some are not going to change activity at all. So it's a combination of the pattern of activity of all these different hair cells in the otolith organs that's going to tell you what direction you're accelerating in terms of horizontal and vertical. It's also interesting that at the striola in both of these maculae, the sensory organs, that orientation of those hair bundles shifts. So the tallest stereocilium is here, but then if you go on the other side of the striola, it's here. So that's kind of interesting. So this shows you one of our maculae. So here we have our otolith membrane. Sometimes those otoliths are also called otoconia. Otoconia just means small rock. And so here we have our otolithic membrane. We have those hair cells poking up into that otolith membrane. You can see that some of them, here's the tallus stereocilia, but then on the other side of the striola, now the tallus stereocilia is that way, so they have different orientations. This is just the location of these sensory bodies relative to those semicircular ducts, and here they are in the otolith organs, which are more central here. And then this shows how a head tilt, so if your head is tilting, this otolith membrane is going to be dragged towards the direction of that tilt, and it's going to activate some hair cells by depolarizing them and hyperpolarize other hair cells so that you know that you're having a head tilt in one direction or the other. And then this again just shows you the orientation of those hair bundles. So this gives you a summary of how the hair cells and the afferent nerve fibers work together. So here we have our otolith membrane. We have our kinocilium, our hair bundle. Here's our hair cell located in the otolith organ. And then here's one of the fibers from our vestibular cochlear nerve. So when our head is straight up and down, we don't have any tilt, we're going to have some baseline activity of these hair cells at rest, some release of glutamate, so we're going to have some activation of the associated afferent fibers. If we tilt our head in one direction, we're going to get some of these hair cells are going to have their hair cells, their stereocilia bent towards the tallest stereocilium, increase glutamate relative to rest, increase the afferent fibering of that vestibular cochlear nerve. Then if we go back up to a straight up position, that's going to go down to a baseline level of activity. And then if we tilt our head in the opposite direction, and we're still talking about that same hair cell or population of hair cells, now it's going to be moving towards the shortest stereocilium. We're going to get hyperpolarization of that hair cell, a decrease in glutamate, and that's going to result in decreased impulses or firing in our associated nerve fiber relative to rest. So that's how they kind of all fit together, and how this system is going to detect head tilts in addition to linear acceleration. So this is to remind you again, here we have just a generic vestibular hair cell. We have the top being bathed by endolymph. We have that gelatinous mass. We have the basolateral part being bathed by perilymph. At rest, there's going to be some glutamate coming out. We can get a change in that depending on whether the hair bundle is being pushed towards the tallest or shortest stereocilia. And then that change in glutamate is going to be detected by glutamate receptors on part of cranial nerve 8, the vestibular component of the vestibular cochlear nerve. Those cell bodies of that nerve are going to be located in the vestibular ganglion. They're still in the peripheral nervous system. And the central part is going to project into the vestibular nuclei, which are located in the brainstem. So this slide is an introduction to our next lecture. Here we have our hair cell. Here's our vestibular cochlear nerve, the vestibular ganglion, and it's going to go into this vestibular nucleus on the ipsilateral side. And what we're going to talk about in the next lecture is how these inputs then and the change of activity in these inputs are going to affect the cells of the vestibular nuclei and lead to changes in extraocular eye movements that are going to go more towards the eye, and also changes in activity in the vestibulospinal tracts and, well, the lateral and the medial vestibulospinal tracts. So we'll talk about those in the next video, the next lecture.